So we can get started now. Welcome everyone to the 2021 Notter Lecture. This lecture commemorates Dr. Mary Notter. Many of you whom knew her. She was a faculty member here at the University of Rochester for many years. And Mary was internationally known for her research in cell biology and growth factors. She received her bachelor's from Penn State in zoology and her doctorate in physiology in 1973. She joined the UR faculty in 1976 and led a successful research laboratory until her death in 1991. Her research studies focused on neuronal growth, differentiation, and development with implications for Parkinson's disease and retinoblastoma. But the story at U of R for Mary Notter doesn't end there, since her daughter, Sarah Notter, whose artwork is featured here, graduated from the U of R Medical School and now practices medicine in Florida. So this named lectureship commemorates Mary Notter as a renowned scientist, a mentor, an educator, and a beloved member of the neuroscience community here at the University of Rochester. As I noted, this slide depicts something besides being a successful scientist. I think it depicts the balance of work and family. And that's something that we find is so difficult as a scientist for many of us to achieve. It's also something that our speaker today knows a bit about and has spoken about. I'm pleased to introduce our Notter speaker.
We need to follow these patients. We prefer if PEDS students don't follow them.
Everybody please mute. I'm pleased to introduce our Notter speaker, Anne Churchland. She's a professor of neurobiology in the David Geffen School of Medicine at UCLA, and she received her bachelor's from Wellesley College and her Ph.D. in neuroscience from the University of California in Steve Lisberger's laboratory. She went on to do her postdoctoral training at the University of Washington in Mike Shadland's lab, where she began her studies of decision-making. In 2010, she became a principal investigator in neuroscience at Cold Spring Harbor Laboratory, where she remained until her recent move to UCLA in 2020. In addition to her research, Anne is a passionate advocate for women in neuroscience and for speaking openly about the balance of work and family and for diversity. She created Anne's List, which is an extensive list of women in the fields of systems neuroscience and computational neuroscience. It began as just a very practical list of women whose work she encountered and who could be nominated as speakers for conferences that she was helping and serving on. Since then, it's grown into a go-to resource for many of us in the field of neuroscience who advocate diversity and gender balance in conferences, panels, and fellowships. She's the recipient of a number of honors and awards, including the Pew Charitable Trust Scholar Award, the Klingenstein Simons Fellowship Award, and importantly, the Louise Hanson Marshall Special Recognition Award from the Society for Neuroscience, which honors individuals who have significantly promoted the professional development of women in neuroscience. Lastly, I'll add her fun fact, which we often do for our talks. She maintains a very popular YouTube channel featuring her research, which she formats for a wide viewing audience. I hope you check it out. It's a lot of fun. So please join me in welcoming Anne Churchland.
Thank you so much for that introduction. It's really a great pleasure to be speaking to you all today, and I'm particularly honored to be able to give the Notter lecture. I'm very grateful for that opportunity, so thank you very much. I used to sometimes spend time at Rochester as part of a collaboration, and one of the things I remember the most is that I would always feel like I didn't have time, have enough time, because there were so many people that I wanted to speak with about their amazing work, and also who were wonderful, wonderful folks on top of that, that I always felt like I was short on time to get to interact with all of the amazing community that you have there. So speaking of work-family balance, I'm giving my talk from home today. I'm part-time back at the lab now because COVID restrictions are getting a little lighter, but I'm here at home. I've got a dog and teenagers. Some of the organisms in this household are grouchy today, so anything could happen. So we'll look at that as an exciting adventure. And who knows what will unfold. Okay. Okay, good. So the starting point for the work that I'm going to tell you about today is that we wanted to understand how widespread brain circuits support decision making. And in my lab, we studied decisions in the context of movements, and that's a natural thing to do for a couple of reasons. And the first reason is that many decisions just give rise to movements, sort of as part and parcel of what they are. So for example, if this mouse here, if it decides that it wants to harvest those blueberries, that will naturally give rise to a rearing movement that will move its head to the right place to acquire the fruit. A second advantage of studying decisions in the context of movements is that it's a really convenient thing to do from the point of view of a laboratory experiment, right? So if an animal is making a licking movement to report a choice, or a human is making a key press to record a choice, then it's very convenient for us in the laboratory to track multiple instances of a decision and also some of the metrics associated with those, like the time it takes to make the decision. But this approach, studying decisions in the context of movements, has led to a known challenge that is familiar to many of you in the audience, I suspect, and that is untangling neural activity from movements from neural activity from other cognitive processes. In our case, that would be decision making. That's the process that we care about, but a lot of the neural activity, of course, is related to movements. This has been known for a long time, and people have come up with some really clever ways to handle the fact that we instruct animals to make movements. I'll call those instructed movements to report choices. So for instance, you might have an animal make a decision before it knows what movement will be used to report it. You might have a 2-interval situation so that the animal doesn't know what the second stimulus will be until late in the trial, so can't plan a movement. Those have both been successful approaches. You might also track a small number of movement effectors, like the animal's head position, in an orienting task, and then kind of regress that out, for instance, when you're interpreting the neural data, so that you can aim to isolate the effect of movements by themselves. So in other words, experimenters have come up with pretty good ways for accounting for instructed movements when they're task aligned, that is, that they occur at a stereotype time relative to events that we as experimenters put into the task. But so in my lab, we started to wonder whether maybe this neural activity only scratched the surface of all possible sorts of neural activity that might occur. So let me be a little more precise about what I mean. So let's suppose that you have a situation like the one I'll tell you about today, where you have an animal that's making a licking movement, maybe to one of 2 choice alternatives, like 2 spouts. And we would collectively call that, as you know from the previous slide, an instructed task-aligned movement. So in the schematic here, I've shown 3 trials, each schematized by a black line with a bump in it that's meant to indicate the lick. And in this hypothetical example, there's a stimulus, say a flashing box, and the lick occurs at the same time immediately after that stimulus and yields a reward at the drop of water. But as those of you who do animal work know, sometimes animals engage in other movements as well, including those that were not instructed by the experimenter. So for example, an animal might make a hind limb movement, or maybe 2 hind limb kicks synchronized to the onset of a visual stimulus, even though the animal was never trained to make those movements. So I'll refer to those as uninstructed movements. And in this example, they're task aligned, because you'll see those 2 hind limb flexions, those two upward bumps in the black lines, occur at the same moment in every trial. Animals might also make task independent movements. That is, they might make hind limb flexions or movements of any part of the body that are idiosyncratic and not related to task events, as I've shown here. And then just to complete the matrix, anyone who has trained human or animal subjects knows that even when you instruct a subject to make a movement at a particular time, there's some variability in when that movement precisely occurs due to the subject's reaction times and a number of other features that I won't have time to go into. So these instructed movements have a task independent component as well. Okay, so what's the big question here? So we set out to ask the following. Do brains prioritize cognition and instructed movements, or do other signals dominate? Okay, so to get at this question, we set up an auditory visual spatial detection task in head-fixed mice. And in this task, there are lights or sounds that are presented on the left or the right. And the job of the animal is to make a licking movement to the side where the stimulus occurs. And I should say that this is a pretty simple task relative to other auditory and visual behaviors that we've developed in the lab. It built on a large body of work that we've been developing over the last 10 years. There's one piece of work that's quite relevant, but I won't have time to tell you about looking at lapses and mistakes made on easy trials that just came out in eLife. I think I'm already losing my voice, so I'm gonna go get a glass of water. So as I step away, please contemplate decision-making and lapses, and I'll be right back. Okay, I hope that was a productive pause. Okay, so I'm now going to show you a video of what the task looks like. So now you're viewing the mouse from below. So that's its nose at the top of the screen. And to initiate the trial, two handles that you see at the bottom of the screen are going to move in. They're controlled by servo motors, and the mouse will make contact with those using its paws. Okay, so that's how the mouse initiates the trial, by making contact with those handles. And now you're seeing the mouse from the opposite point of view, this time from the back. Excuse me, you can see its tail hanging down there at the bottom. And now you're going to see a visual stimulus on the right. It is presented twice with a little gap between the two stimulus presentations. There's then a 1 second delay where the animal has to hold its choice in mind, and then it's allowed to make a response. So during this period, you're going to see both spouts move in. The animal will make a choice for one spout, and the other one will move out of the way. And that's to prevent the animal from changing its mind. Okay, so that's how animals report choices. So as you might infer from my showing you these videos, we collected a lot of video data in the course of measuring the neural activity in this study. We had 2 cameras positioned on the animal's face and on the animal's body. And this allowed us to do a couple of things. First of all, to track particular movements of interest, like the pupil diameter, which you can see expands and contracts in rather complex ways throughout the trial. And it also allowed us to use this video data in an unsupervised way that I'll tell you about more in a moment. Okay, so remember that our goal is to determine whether cognitive or movement signals dominate neural activity in experts, that is in animals who are trained to do this task. And because of this, we felt the need to look at cortical activity really quite broadly. We didn't really know what would be the right place to look. We were sort of open to the possibility that there might be interesting signals in many parts of the cortex. And so we decided to use a technique called wide field imaging. So we start with a mouse that is expressing calcium in all excitatory neurons. So for those of you who are not experts, that means that when a neuron fires, it turns green and emits photons. And we can capture those photons with a camera, a CMOS camera, that's part of a wide field macroscope that we built in the lab. So we illuminate at 2 different wavelengths, 473 nanometers, and that's to activate the GCamp so that we can see the neural activity. And then for aficionados in the audience, we also do a lower wavelength illumination. This is violet light, and this is so that we can correct for hemodynamic activity. We subtract that out so that we can isolate the calcium activity. This is turned out to be a pretty useful preparation, and it's also pretty easy and cheap. It's a lot easier to make than a 2-photon microscope, and it's a lot cheaper. And as you'll see over the course of this talk, it gives you this interesting kind of bird's eye view of what's happening in the cortex. It also has some downsides, which I'll tell you about. But if you're interested in building one yourself, I highly encourage you to do so. And we have a protocols paper that is just in press in Nature Protocols that has all the details. So I'd encourage you to build your own if you want to try these experiments at home or otherwise. Okay, so the first advantage of this approach is that we can image activity through an intact skull. And this is great. This is, you're seeing here the mouse brain. You can see all the vasculature really beautifully. And this means that the experiments are pretty high throughput. We don't have to worry about implanting a window and keeping the window clear, infection, and so on. We can just image through a skull. This means we can do lots more animals than we would if we had to do implants on all of them. And it also means we can image longitudinally, which is a really big win. The other nice feature of this approach is that we did this in the mouse, and the mouse cortex is lysencephalic, which means it's flat. And that means we can easily visualize multiple maps in multiple visual areas. So to do that, we have a passively viewing mouse view horizontal bars that move vertically and vertical bars that move horizontally. And then by looking at which phase of that stimulus each pixel is responsive to, we can, excuse me, we can generate retinotopic maps that include primary visual cortex and usually about 5, sometimes more surrounding visual areas. So this is a big win. First of all, because it allows us to confirm that the technique is working because we see maps in the right spot that have the right visual field sign. And secondly, it allows us to use this along with some other skull landmarks to align our activity to the Allen Institute common coordinate frame. And this is really useful because in the back of the brain where we have the sensory cortices, we can make maps to pinpoint where they are in individual animals, but it's much harder to be able to pinpoint areas like the frontal cortex, which are not as easily mapped. So being able to use a combination of the visual field maps and also the skull landmarks to align to the Allen Institute common coordinate frame is a really, really useful technique. So this is always the first step that we do in all of the animals. In the work that I'm going to tell you about today, there are 11 animals included, and you can see they all have nice visual maps, often on both sides, although we only show the stimulus contralaterally. And so once we've established in each animal that we can see good maps and that we're getting reasonable signals, then it's time to go back to the decision-making experiment that I told you a moment ago. So I'm going to show you now just some raw data. So you're just going to see the video from the 2 cameras and also the fluorescence activity on the right. And you're just going to see a number of trials go by, and they're slowed down by, I forget, about a factor of 5 or so. And so as you do this, you'll be seeing the data really as we did for the first time. We just kind of sat there and just looked at it and just saw what was there. So a couple of things were really striking to us as we sat and looked at the data. And the first thing was that the activity was quite widespread. This is a pretty simple task, right? You just see lights on the right and lick to the right, or with sound similarly. And yet it seemed that this relatively simple task was recruiting neural structures very broadly across the dorsal cortex. That wasn't something we necessarily expected. The other thing that really jumped out at us, the second thing, is that the activity was highly variable trial to trial. And again, this seemed kind of surprising because the animal is really doing a fairly simple sensory motor task. It wasn't obvious why such different neural structures would be recruited on every trial. So we pondered this, and we at first were, of course, skeptical as to whether we were getting reasonable signals at all. So the first thing we did is said, okay, let's forget about single trial activity for a while. Let's go back to our old friend, the mean, and make sure that the mean activity that we're seeing is reasonable. So what you're seeing here is what we call delta F over F, the raw fluorescence signal. It's averaged across all trials for all visual stimuli and all subjects. So this is like a super grand average. And we average left and right trials for any vision aficionados in the audience who are wondering why V1 is lighting up on both sides of the brain. And you can see here, as you look at the neural activity for different moments in the task, that it's pretty broadly more or less what you would expect, right? So when the visual stimulus comes on, you get activity in V1 in the back of the brain. In the delay, things are a bit quieter. During the response period, there are multiple sensory and motor areas that are activated. So this was kind of encouraging. We decided to go dig a little deeper in the mean activity in area V1, just because that's an area about which the response properties are very well established. So what I'm going to show you here is delta F over F. That's the standard way for measuring the overall fluorescence signal. For V1, for stimuli, I'll show you the neural activity in V1 for trials where a visual stimulus was presented versus an auditory stimulus. Hopefully there's going to be a big difference. This is time on the horizontal axis, and here it's aligned to the handle grab, so it's before the stimulus comes on. And you can see, just as you'd expect, when the stimulus comes on, that we have a big difference in V1 for trials with lights versus tones, and that they obey the proper temporal dynamics that you would expect from primary visual cortex. And the responses mostly come back together again at the end of the trial. Great, so we're seeing the right signal in the right place at the right time, very reassuring. We then did the same comparison for left versus right stimuli. And as all of you know, if you're measuring in left V1, you expect to see a higher response when stimuli are presented in the contralateral hemifield, that's the right. And that's exactly what you see here. The gray trace is above the red trace, so we see a larger response for lights on the right relative to lights on the left. Great, so this was encouraging to us, being new to wide field imaging and really to calcium imaging in general, reassuring that we were seeing the right signal in the right place at the right time. So then we said, okay, let's go back to this single trial activity, which was bizarrely widespread and surprisingly uh-oh. Okay, I think we're back to normal there. And see if we can start to make sense of that single trial activity. So what we wanted to do was this. We wanted to say for any pixel in the brain, that's not quite right because we reduced the dimensionality of this considerably, but stick with me for the moment. For each pixel in the brain, let's suppose this is the activity on the fluorescence activity on trial 1 and trial 2 and so on. And what we'd like to do is be able to build a model so that we can predict the activity on single trials and capture all of these fluctuations with our model. I should say when we set out to do this, I was rather skeptical that it would work at all. And the reason is because I've studied fluctuations in neural activity for many years. The brain is notoriously variable, and much of that variability we think of being something that you can never recognize. The brain is stochastic. It's just different from trial to trial. So this seemed to me like a bit of a fool's errand to try to capture all of these fluctuations. But the postdocs in my lab, this was led by Simon Musil and Matt Kaufman, they were adamant that, that this was a reasonable enterprise. And this is what they said. They said, look, we have a lot of really interesting environmental variables, task variables, and so on, that might potentially explain these fluctuations. So for example, the stimulus comes on at a different time every trial. That's by design. The handle grabs and licks are at different times on every trial, and that's because those are under the animal's control. And so they just do them when they want to do them. And finally, they pointed out, look, we collected all of this video data. Let's just use this video data. We don't know how to use this. Let's just take it. We'll reduce the dimensionality a whole bunch, take, I don't know, the top 200 dimensions. And then let's just include those in a model as well to see if there might be some information in the video that we haven't thought of that might be important for explaining neural activity. So we took all of these behavioral and movement variables together and put them into a big design matrix, and then aimed to fit a model so that we could predict the trial-to-trial fluorescence using the regressors that I just described a moment ago. And we did this using ridge regression to prevent overfitting. So I'm going to have just a brief technical digression here for one slide. If you're not into technical stuff and model fitting, you can zone out for one slide and I'll tell you when to come back in. So you might notice that in the regressor column, there are really two quite different sorts of regressors. So these at the top are event-based. For these, the model learns what we call temporal event kernels. And the other kind of regressor are what we call filterless analog predictors. There's things that just unfold continuously in time. So these temporal event kernels, for these, the model can learn any sort of time varying response. This is the one for V1 that the model learned from our data. You can see it's really complex and time varying, even though the visual stimulus comes on at a precise moment in time. And these have been used pretty extensively recently in accounting for neurophysiological data. And really this approach was borrowed from fMRI, where it's used really quite extensively because it's similar to our situation. There are many different neural signals that are measured and they have varying time courses. So this other kind of regressor, the filterless analog regressors, these are really useful, excuse me, when the analog, when the regressor is like a scaled version of the fluorescence, right? So if the two of them are kind of going up together, these regressors totally kick butt, but they're much more limited if there's a temporal gap between them or if there are complex dynamics in one signal but not the other. So they're not really very flexible in terms of capturing complex dynamics the way the temporal event kernels are. And I mention these because the video data is, of course, an analog regressor. It's not something that occurs at a particular moment in time in an event-based way. It's just unfolding continuously. Okay, so if you tuned out, then it's time to tune back in because I'm going to tell you how the model did. Okay, so we built the model with all these regressors, and I'm now going to show you how the model performed. So if you look over at the trials on the right, I'm going to overlay the prediction of the model on held out trials. So it's not a fit, it's a prediction for data that the model never saw. And that's what you see now in red. And when we first saw this, we thought it wasn't bad. We were actually accounting for a lot of these fluctuations that seemed at first that they would be utterly mysterious. So I know this crowd is not going to be convinced by 3 example trials. So I'm going to show you now the performance of the model over the entire brain at a couple of different moments in time. So the metric that's going to be important for the moment is what you see on the left, CVR squared, and that's short for cross-validated R squared. That means how much of a correlation there was on cross-validated trials versus what actually happened in the brain. And red means, or yellow means the highest correlation. So you can see this is pretty good. We were able to capture about 42% of single trial variance, which we were really happy about. And this wasn't just restricted to a particular part of the brain, although there were hotspots, or to a particular moment in time, we were able to explain variance really across the whole brain at all moments during the trial. So this is really encouraging. So he said, okay, great. Well, let's look under the hood of the model and see how this is working. So what we did is as follows. We looked at how much variance was explained by each of the regressors that were included in the model. And I've divided these up into 3 groups, which I'm going to be using for the remainder of the talk. You should feel free to dispute these groups. They're somewhat arbitrary, but we did this sort of for a reason that you'll see as the talk goes by. But again, feel free to push back on this. Okay, so the first category of regressor are things we call task variables. And these are things that any sort of self-respecting decision-making lab would include in its study. Things like the moment that the sensory stimulus comes on, whether water was given, choice on the current in previous trials, and so on. And you can see that these account for some amount of variance, some more than others. Okay, these are now the instructed movements. Remember, I mentioned these at the beginning of the talk. These have been studied extensively. It's the impact of movements that we train the animals to make. And it's a good thing that people studied these extensively because they account for quite a lot of variance in the neural activity. Not really news, but a nice reassurance. Here was the thing that was surprising. The majority of the variance in the single trial neural activity was accounted for by uninstructed movements. Those included the pupil, hind paw, nose, body, whisking, the raw video data, and the first derivative of the video data, so how it's changing over time. We did not expect to see this. So we went back to the data and said, okay, let's look at what's really happening here, because it seems surprising that these uninstructed movements that had nothing to do with the task at all would account for so much of the variance in these trained experts. So we went back and looked at the data. And at first, we immediately realized that something slightly funky was kind of going on here. So this is showing you the spatial kernel, that is the contribution of each pixel in space for the regressor, the onset of the visual stimulus on the right. Okay, so you can see that we see a big hotspot in the contralateral V1, the left V1, that's great, just what we expect. But then there were some weird like hotspots out here. And it was not immediately obvious to us why we would be able to explain variance in these more anterior parts of the brain based on the onset of the visual stimulus. That was a bit weird. Okay, 2 other weird things. So this is the variance explained by a right handle grab. Mysteriously, we saw very symmetric activity on the left and the right, which seems a little odd given what we know about motor control. And then finally, this one's really weird. This is the variance explained by the nose movements. Well, remember that the olfactory bulb is up here, and it really pales in comparison to these 2 kind of baseball-shaped areas, which encompass both somatosensory and motor area in more posterior parts of the brain. So this kind of tipped us off that something was a little bit weird and that perhaps we had not had a sufficiently conservative approach in the model. So we went back to the model and we said, look, we really need to see the contribution of these regressors one by one, because we really want to put the feet to the fire of this possibility that uninstructed movements are accounting for so much neural activity. So we said, great, let's go back. We're going to kick out the regressors one by one, and then we're going to see how much less variance the model can explain. So the intuition, which is probably obvious to many of you already, is that if 2 variables are correlated and you eliminate one from the model, you don't change the overall ability of the model to explain the data. So we did this for every regressor and then refit the model and redid the cross-validation. And we did this for all of the regressors in the model and cleaned things up a lot. So now, instead of having the onset of the visual stimulus explain variance in anterior areas, it's very restricted to V1. Similarly, we now see a nice contralateral, we see pixels in the contralateral hemifield able to explain variance for the handle grab. And then this one was really, really profound. So once we did this experiment in the proper way, we saw that activity for the nose was greatly spatially restricted to the olfactory bulb. So why did this happen in the first place? Well, what the difference between the top and bottom row, what this tells us is that there are many movements that are happening at the same time, right? So you can think that the visual onset explains variance in the anterior part of the brain, but that's just because there are other regressors that are occurring at the same moment as the onset of the visual stimulus. And I think the other take home that I hope you will remember is that you sometimes see people looking at wide field activity or other kind of metrics, measurement modalities that give you a whole brain picture. And they say things like, oh my God, the whole brain lights up. Well, yeah, the whole brain lights up, but that's because there are many things that are occurring often at the same time. So if you really want to know the contribution of a particular variable that you care about, you have to really be careful to kind of pinpoint its contribution directly. So we did this for all of the variables in our study. You could look at this plot for a long time. We may have to bring it back up in the discussion. But looking at this more conservative metric, which we call cross-validated delta R squared, right? So it's the change in R squared. It really cleans up the signal a lot. So we could then go back and look not only at the raw cross-validated R squared, but the delta R squared, and to see whether this dominance of the uninstructed movement persists. And just to give you one final intuition, we sometimes refer to this metric, this delta CVR squared, as kind of the suffering index. It's how much does the model suffer when you remove a particular variable and don't allow that to be included in the model fit. So naturally, we expect these values to be much smaller because they're the change in the model performance following the omission of that particular variable. Okay, so you can see that these task variables, I mean, they're important. They account for some unique variance, but they're quite small compared to instructed movements and even more so compared to uninstructed movements. So for the remainder of the talk, I'm gonna mostly talk about these different variable groups, although feel free to ask me if you wanna know about individual movements and how they were impacted by these analyses. And then just to summarize for these three variable groups, for both the group model and the unique contribution, that's the more conservative method, we found that uninstructed movements just absolutely dominated the neural signal. So this is a highly rarefied plot. A lot of things are compressed in this plot. I'm going to first unfold this over time. This is important because you might wonder whether uninstructed movements dominated because they're supporting the instructed movements, right? Maybe when the animal is licking, it sort of pushes itself forward, and these the movements like pushing on the hind limbs might support the instructed movement of the lick. But in fact, that's not the case. So the uninstructed movements dominate throughout the entire trial, including the pre-trial interval when the stimuli have not even started yet. And if anything, the contribution of this variable group tends to dip around the time of the licking and stimulus onset and so on, mostly because the instructed movements have a bit of an uptick at that time. So now I'll just unfold this in space and time, which would be a movie. So now you can see the delta CVR squared for every moment in time and every moment in space. The stimulus comes on briefly. Primary visual cortex has its moment in the sun. But for the remainder of the trial, the uninstructed movements dominate. And even when the stimulus is on, the contribution of the task variables is still smaller than the contribution of the uninstructed movements, even in V1. Okay. So I want to just take a step back here, and because I think these conclusions to us were surprising, and I want to point out that our approach differed from previous approaches, not only in the size and diversity of movements that we included, but also in the fact that we sort of we allowed for the possibility that any movements in the video might matter, right? We had a very unsupervised approach here, in that we just took the video, we squished it down and took the top 200 dimensions and just threw it into the model and kind of let the model have at it. And the upside of this approach is that we learned something that to us was really surprising, that these movements turned out to matter a lot more than we thought they would. That's the upside of the unsupervised approach. The downside of that approach is that one is really left without a convenient low-dimensional summary of what actually happened, right? We don't really know what the animals did, right? We just know that stuff in the video mattered. So one way to start to relate the neural activity to the video is to look at the weights that were fit by the model. So we can take those weights and we can project them back up into the full dimensional space. And that's what you see here. And we call this the hot mouse because each pixel is color-coded according to its contribution to the fit of the model. And this is sort of vaguely informative, I suppose, right? We see that there are pixels around the animal's jaw and eyes that seem to matter. Perhaps that makes sense, right? We've learned from recent work that came out over the summer that facial muscles are really important for showing emotion. So it may be that these facial expressions are informative, but the truth is that we don't really know, right? We know that these facial movements matter a lot for explaining neural activity, but it's hard for us to understand more about, it's hard for us to know at this time more about what these movements actually were and how they fit into how we think about decision making. We're trying to get to the bottom of that, and maybe I'll tell you about that next time I give a talk, because we're still working on it. And I have more to say in the discussion. So if people are curious about more about what the movements were, I'd be happy to return to this point. We had a cosine poster just last week that dug deep into this. Another approach that we're taking is we're working with Liam Panitsky's lab and Scott Linderman's lab, looking at new methods to relate behavioral video to neural activity, going through tools that kind of segment behavioral videos into behavioral syllables. So I think that's kind of a promising approach. And I think overall, this approach kind of highlights a change in how one is able to go about questions in systems neuroscience. I think in the past, labs like mine were really tied to what you might call a task-based approach, right? We train an animal to do a behavior, we track the relevant metrics that we've taught it to do, and we try to use those to explain neural activity. And historically, this has been kind of separate from what you might call the observational approach, right? Where animals are wandering around on a wheel or in a maze, or they're exhibiting spontaneous behaviors that are then tracked by the experimenter. And I think in the past, one had to sort of pick and choose between these two extremes, right? You had to either fully go to the task-based psychophysics or you were fully in the observational. And I think now these new methods for video processing, automated video processing, mean that we don't have to choose anymore. We can kind of go back and forth between this task-based approach, which has many advantages, and this observational approach, which tells us about the spontaneous movements that the animal is electing to engage in. So I think it's a really exciting change in the field, and I think it's going to mean that we can not only take advantage of these really powerful tasks that are informative about computations, but look at them in the context of many other behaviors that the animal has decided to engage in. And I think, sort of just as a brief aside, we can further understand these neural dynamics by making really good cognitive models to estimate latent variables, things like how much evidence is accumulated or how biased the animal is. And we can also gain further insight from artificial organisms that we can train to do the task that might have similar or perhaps different strategies. And they may be similar or not to the biological organisms. And I think this will allow us to get around a big problem that many of us who do animal work and human work as well know, which is that sometimes individual organisms can have very different approaches to solving a problem. So in this image, which is from a review that I wrote with these folks below, these animals all have the same goal, which is to get the cheese, but they're going at it in very different ways, including an artificial organism that has a fourth way altogether of trying to solve this problem. And in the past, this subject-to-subject variability was sort of intolerable because it was very hard to characterize. But now we can characterize what all of the individual organisms are doing. And instead of that being a nuisance, it can be kind of an inroad into understanding variability across animals that previously made neural data really difficult to interpret. So I think a first way that we're trying to do that, to sort of more deeply understand neural activity and to connect it to behavior, is by looking at what happens not only to behavior in its final expert form, but in the transition from novice to expert behavior. So to do this, we took advantage of the fact that imaging through an intact skull means that we can image over many days and even over many months without having any compromise in signal quality. So we did this with a number of animals. We looked at we tracked their brain activity all through this training period, which was about 45 days. And we looked at how the relationship between task and movement variables evolved over learning. So on the vertical axis here, this is the cross-validated R squared. And now on the horizontal axis, instead of time in the trial, this is now time during training. So this is many days. And I'm going to show you the contribution of each of these variable groups over this many-day training period. And these are the same groups that I've been talking about throughout the seminar. Okay, so a lot is happening here. So the first thing is that there's an increase in the contribution of instructed and task movements as training unfolds. This makes sense. Originally, when the animal is encountering these sights and sounds, they're utterly meaningless to the animal, but they come to have meaning over time as the animal learns that they provide an instruction for what it's supposed to do. The other thing that's happening over training is happening to the uninstructed movements. The first thing to notice is that they always dominate. So they always account for more of the single trial variance than either of the two trial groups. But notably, their contribution does decrease in a very real way over training. And so we wanted to understand what was happening there. So we had two hypotheses. The first thing we said, well, maybe the reason that the uninstructed movement contribution decreases is because the impact of individual uninstructed movements changes. Okay, so what would that mean? That would mean that the same forelimb flexion had a big impact early in training and a smaller impact later on. And there's lots of reasons that that was a candidate hypothesis, right? It's often the case that you see cortical modulation of particular activity that later leaves the cortex as animals become experts. So to get at this, we just went back to the weights in the model again, and this time looked at how those weights changed over the many-day training period. And we did this for, I think, 5 animals here. So this is just the normalized beta weight. That's the contribution of each of these variables over training days. And not much happened really. It's pretty stable over time. So it seemed like basically a given movement, it kind of packs the same punch, whether it occurs on day 1 of training or occurs in an expert. So it wasn't the case that the contribution changed. So then we said, okay, maybe it's something else. So we asked, does the frequency and timing of uninstructed movements change? And to make this clear, I'm going to show you an individual movement. This is whisking. So this is whisking early. It's the number of events as a function of now time in the trial. Okay, so it's not training time on the horizontal axis. We're now back to very brief trial time. And this is not a very interesting plot. It basically tells you that the animal is equally likely to whisk at any moment in time. And this comes from an animal very, very early in training. Late in training, totally different story. So late in training, there's a very stereotyped whisk at the moment the visual stimulus comes on, and another slightly less stereotyped whisk around the time of the reward. So remember, whisking, as far as we know, has nothing to do with the trial, right? We didn't train the animal to whisk. It's a visual task that you report by licking. But for whatever reason, for the animal, this became an integral part of doing the task. And so sort of to put this another way and to kind of harken back to nomenclature that I introduced at the beginning of the seminar, the task aligned contribution of these whisks went way up, and the task independent contribution went down. It decreased considerably. So what do these concepts really mean? Task aligned and task independent. How should we think of those in the context of movements that humans make? Is this even reasonable to think about going from animals to humans? Well, a task-aligned uninstructed movement would be, these are actually seen very frequently in humans, and they're especially seen in humans that are very well trained to execute a stereotype movement. So this is Nomar Garciaparra, who's a professional baseball player, plays for the Boston Red Sox, and every time he goes up to bat, he has a stereotyped set of movements that he makes before he hits the ball. I'll show you that one more time. And from our point of view, it's not obvious how these movements relate to hitting the ball. But from his point of view, clearly they are relevant because he does it every time he gets up to bat. Here's another baseball player, Mike Hargrove, who has a very nice task-aligned uninstructed movement. He hits his feet twice with the baseball bat. And these movements are pretty cool. They've been noted and studied previously in both humans and in animals. And I would presume that the reason these occur is because of reinforcement learning, right? If you have a successful outcome, like hitting the ball, your brain doesn't necessarily know which of the preceding moments were critical for that success. And so many of them are reinforced, even ones that seem like they're probably not relevant to the success of the well-learned movement. I think perhaps even more interesting to me are the task-independent uninstructed movements. And in fact, humans make these all the time: leg shaking, pen clicking, hair twirling, fidgeting with eyeglasses. These are very, very common movements that people make. They often make them, especially when they're trying to concentrate. If we were in a seminar right now, you could look around and see everybody fidgeting because people do it all the time. People also do them despite the fact that they have a metabolic and also a social cost. Actually, people get really angry when people fidget, which is sort of bizarre. And yet these movements persist. So we don't know the function of these movements in either humans or in animals, and when they help and when they hurt. But they certainly are movements that are common across species, and their persistence is surprising and interesting. Okay, so I'm going to address just a few potential concerns that you might have. The first thing is you might wonder whether this dominance of movement, of uninstructed movements, is this only evident because we measure a signal that's pooled over many neurons? So a downside of wide field imaging, of course, is that it's not a single neuron measurement. We're seeing the pooled activity of many, many, many neurons from many different layers in the cortex. And so one way that we got at this issue was to go to a single cell method where we could track individual neurons. And we took advantage of the fact that we had recently brought 2-photon imaging as a new measurement modality into the lab. This was led by Farzaneh Najafi and Matt Kaufman. Farzaneh led a really interesting project about inhibitory neurons and decision-making models, which is published. I'd encourage you to look at. And we were very fortunate to have this set up, and we're able to measure single cell activity using the 2-photon in 5 different brain areas. So these are medial motor cortex, anterolateral motor cortex, primary somatosensory and visual cortex, and also retrosplenial cortex. We used an automatic segmentation method, SUITE2P, to identify individual neurons. And before looking at the contribution of these neurons to instructed and uninstructed movements, we first verified that the trial averaged activity was doing what it was supposed to do and was similar to known responses that have been reported extensively in the literature. I skipped a whole slide here. Okay, hey, we'll go back to the discussion. I'll show you the data. So basically, we found really similar findings with the 2-photon versus with the wide field, and that was an accidental omission. So remind me in the discussion to show me the data. You shouldn't believe what I'm saying without seeing it. Okay, so the second concern that you might have is that you might worry that these responses that I've mentioned are restricted to the cortex and are not true in deeper regions. So to get at that, we took advantage of the fact that we were measuring neural activity from multiple cortical areas using NeuroPixels probes. And this was a project that was led by Ashley Joavinet when she was a postdoc in my lab. And she was measuring neural activity in freely moving animals using NeuroPixels probes, and also had built a really nice implant so that after recording activity in freely moving context, she could then head fix the animals and show them traditional visual stimuli in a more controlled setting. So she measured activity from many different areas in both cortical and subcortical. And it was in a different task, passively viewing task. So she was able to compare the stimulus-driven activity and spontaneous movement activity. And so she compared the contribution of the variable groups in visual cortex, superior colliculus, and also the midbrain reticular nucleus. And in all cases found that the cross-validated R-squared was really driven by movements and not by the stimulus. And it's really interesting to do the single cell version of the wide field experiment that I've been talking about, because you can look at the level of single neurons and look at single neurons and compare the contribution of movement versus task variables. And neurons are highly idiosyncratic in how they reflect these combinations. So here we have all of the neurons in her study, and they are ordered by the contribution to the movement model. And then in green, you see the contribution of the stimulus model. So for this neuron here, you can see that there's an enormous stimulus contribution. So this is really sort of a traditional, more sensory neuron. This one over here has both movement and stimulus contributions. And this one is really a movement dominant cell. And this turned out to be a very helpful observation for us in understanding these movement-driven responses and also their impact on things like neural correlations. It's important to note that neurons can be side by side, but still reflect really idiosyncratic combinations of stimulus and movement activity. Okay, so before I stop, I'm just going to tell you a couple of future directions in the lab. I hope I have convinced you that movements dominate neural activity, and I think that the 2-photon experiments were really needed because there are many disadvantages to doing wide field imaging that make it hard to interpret. And so you might kind of wonder, well, what really is the advantage of wide field imaging at all? And there's a few things. So first of all, it's really nice to have this big bird's eye view and kind of tells you where you should or might wish to zoom in with a single cell method. But the other big win of wide field imaging and what we're really doing now in the lab is that it allows you to image in a cell type specific way and get a whole cortex view of the contribution of different cell types. So one of the things that I came to really appreciate during my time at Cold Spring Harbor before I moved to UCLA was through a collaboration with Josh Wong, who really highlighted to me the importance of understanding diversity of pyramidal neurons in the cortex. So we think a lot about diversity across inhibitory neurons. That's well known and well established. There are a million papers about PV neurons, but it's much less appreciated that there's enormous pyramidal cell diversity as well. And cells differ in terms of their laminar position in the cortex, their morphology, and also their developmental lineage. And what I came to appreciate working with Josh is that there are developmental factors that play a key role in determining where a neuron ends up in the cortex that you can track and leverage later on in development and use it as a way to identify or even control cells optogenetically. So this is a really new direction for the lab. We're still working on this, but we've been focusing on two different pyramidal cell types, and they're both defined by their developmental lineage. These are both FESF2 and plexin define how the cell gets to the cortex in the first place, and also where it projects to as an adult. So neurons that express FESF2 project what we call corticofugally to many subcortical targets, and neurons that express plexin project both corticocortically and also to the striatum. So these are quite different groups of cells, both in terms of their developmental lineage and also their projection target. And we can take a mouse that expresses calcium indicators only in these cell types and then compare patterns of activation across these different animals. So this is data from a FESF animal, and this is showing you the movement contribution and the task contribution. So this is the same kind of analysis I've been telling you throughout the talk. So first of all, we replicated our finding in these neurons, which was exciting, especially because their cell bodies are very deep in the cortex. We again found that movement signals dominate. And we also were able to compare the pattern of explained variance for these two different cell types. And we found that they were rather different. So we found that in the FESF animals, task variables were mostly restricted to anterior parts of the cortex, whereas for plexin neurons, the contribution was more widespread and distributed. Similarly, we tracked the neural activity of these animals all across the learning process and found that this difference between FESF and plexin animals, the sort of localized versus more widespread, that tended to emerge rather late in training, suggesting that it might be that the difference between these two cell types might be experience dependent and might reflect the expertise of the animal. We're also continuing to dig deep into the contributions of inhibitory neurons. This is a new project in the lab where we have calcium indicators in all neurons, but are able to distinguish somatostatin positive neurons from other classes of inhibitory neurons. And this is work led by James Roach in my lab. So I think in our future directions, we're really trying to understand how these observations that we're making depend on the contributions of different cell types, ultimately hoping to understand how these groups of cells work together to implement the computations that are necessary for decision making. Okay, so I will stop there. So I've told you that cognitive experts engage in diverse uninstructed movements with profound consequences for neural activity, that robust movement representations are widespread in cortical and subcortical structures, and that learning introduces dramatic changes in the timing and frequency of these movements. Okay, so these are the people in my lab that did the work. This project was led by Simon Musil, who you see on the left. He's now a professor in Germany, also Matt Kaufman, who's professor at University of Chicago, and Ashley Joavinet, who's a professor at UCSD. And these are the remainder of my lab members when we were still in Cold Spring Harbor a few months before we moved to UCLA. So we're also really grateful to the people who funded this work and to all of you for listening to what I had to say, and I'd be happy to take any questions. Thanks.
That was great. Thanks, Anne. Did we have a question from the audience? And
yeah, I've got one.
Okay, and if others would like to throw something into the, let me know and I'll call on people from the chat in order. So go ahead, Brian.
Dr. Churchland, that was an excellent talk. My name is Brian Ivey. I'm a first-year neuroscience grad student. I'm in Dr. Huxton's lab in the Center for Visual Sciences. I'm really curious as to how the changes in task-related versus unrelated cortical activity impact task performance and success. And also, what can we do to sort of increase task-related brain power or cortical activity related to the task if success is impacted? And then lastly, how does this sort of activity/recruitment relationship change in injury?
Oh yeah, those are all great questions. Okay, so I will show you. Okay, so to get at the issue of how these movements impact performance, we're still kind of working on that because it's turned out to be a rather difficult thing to get at. But we decided what we wanted to do was to take a more supervised approach to get at this issue. So we started to track individual movement effectors using Deep Lab Cut, and we tracked 25 points, I think, over the course of the trials. And then this doesn't quite answer your question, but it's important. So the first thing we did is we took the tracked labels and asked whether we could decode various aspects of the task. So we said, can we just look at these labels and figure out what the animal's choice is, whether or not it will get rewarded, what its previous reward was, and so on. And we found that we could do that pretty successfully. So this shows you on the horizontal axis, that's the ability of the video data to predict these various quantities, and then it's trial time on the horizontal axis. So you can see in red, that's the ability of the video labels to predict which side the animal went to, which is really good at. Also, we're also able to predict whether or not the animal will get a reward even before the animal has made its movement. And we can also predict what happened on the previous trial as well. So this was kind of interesting. There certainly has been previous data showing that you can kind of guess what an animal is going to do before it does it by looking at things like its eye movements and so on. But we were still surprised to see that there was this much information in the video data. So we then took it a step further, and this will answer your question more directly. So then we said, okay, so let's now flip what we did in the previous slide. Let's now try to predict the video from a whole bunch of choice parameters. So we're going to try to account for the spatial position of these labels as a function of the stimulus, the choice the animal made, and so on.
Muss
Okay,
mal ganz kurz schlitten. Der Papa muss vielleicht eine Frage stellen.
So we do that, and we can account for, some amount of variance that we can explain of those video labels. And then we say, okay, let's look at the variance that we couldn't explain. And we call that task independent variance. It's sort of the remaining variance. So they're, in other words, movements that have like zero to do with the task whatsoever, as far as we know. Okay, so then what we did, and this is the plot on the bottom, for many, many trials, thousands and thousands of trials, this is a single animal, we plot that task independent variance in blue. And it kind of like, I don't know, fluctuates, whatever, goes up and down. And then we plotted alongside that the animal's correct rate, so its overall performance. And this is a pretty reliable effect. We saw this very robustly in 6 of 7 animals. We find that those two tend to be inversely correlated. So as the animal is making more task independent, has higher task independent variance, its correct rate tends to go down. So the more you're fidgeting in a way that has nothing to do with the task, the worse your performance is. So this suggested to us, and we're trying to dig down in this a little bit deeper, that this task independent variance might tell us about the animal's overall engagement level. So we're hoping to kind of relate that to other measures of engagement, like pupil diameter and so on. So the take home might be fidgeting is bad. But I'm not sure. It's preliminary evidence. And your question about injury is a great one. And I also would like to know a lot more about how this is related to different kinds of disease states. So for example, there's a huge variability among people in terms of how much they fidget and move around. And there are populations of people, like people with ADHD, who tend to fidget more. And those movements are a characteristic part of the ADHD diagnosis, and they're also treatable with methylphenidate. But their role is not really understood. And I think it would be great to understand what these fidgets, these movements are doing in different populations of people, hopefully, potentially using animal models as an inroads to understanding that.
Thank you.
Okay, John Mink has a question next.
Really fascinating talk. I'm very interested in the suppression of unwanted movements, and particularly interested to see, as during the training, the uninstructed movements decreasing. And I think a couple of things. You were just talking about fidgeting. I would imagine that fidgeting is more of a problem if you're fidgeting with the part of the body you're going to use to do the task. Mm-hmm. And so I wonder if during training, what you actually see is in the forelimbs, a diminution of those uninstructed movements, and actually maybe a pause in motor activity in those limbs right before they make the movement, whereas tail movement, high limb movement probably doesn't play much of a role. And so I'm wondering if you see, again, a somatotopic change in whether you have movements persist or decrease in a stereotype task-related way, but a decrease rather than a generation of movements.
Yeah, that's a great question, actually. And we haven't really looked at that, although we easily could, right? Because we already know the overall contribution of the uninstructed movements and how that changes over training. So we should break that up on a movement by movement basis. And it'd be really interesting to see whether it's a homogeneous or a heterogeneous, as you propose, change. It's a great suggestion. Thanks.
Okay, we have another question from Marissa.
Hi, Anne. This was really a fabulous talk. I loved seeing the videos, watching the behavior and seeing the outputs in the models was really great. When I think about task-independent movements, I'm always sort of thinking about the stress response. Because stress is really sort of the metabolic supportive behavior, and even giving a talk or beginning to give a question after a talk, you start feeling your heart race and you feel yourself twitch a little and your legs sort of twitch. And even in talking to professional athletes, like some of the reasons they do those movements is sort of help with the stress of the moment. What do you think about sort of habituation? And of course, in like novel contexts and social contexts, there's reasons to have stress HPA axis activation. So what do you think about this idea, maybe what the role the stress response could have, or in the decrease across time, maybe reflecting some sort of habituation?
Yeah, that's a great question. I wonder, one way we could maybe look at that is to ask how the total kind of magnitude of movements changes over a session, right? Because it might be reasonable to think that the amount of stress would be higher earlier in the session compared to, say,
or 20 after a few sessions, yeah.
Yeah, there might be two relevant timescales, right? Because it may be that within a session, stress goes down because the animal's sort of recovering from the stress of having interacted with a human, which will be a very recent memory early on in the session and a more distant memory later in the session. So that timescale might be relevant. And then, as you say, the overall training timescale might also be relevant. It may be that they're more stressed early on than later. We do really try to habituate them to the rig, so they spend a lot of time. First, they have like a whole week where we just handle them and play with them. And then we slowly put them in the rig and give them water. And then it's like after that we start doing the task. But it would be a really easy thing to check, right? To see how these movements change on those two different timescales, and to see whether stress is playing a role. We can get a hint of that at the plot you're looking at now, because each of those vertical lines is a session. So you can look at how the task independent variance changes over the session, like by looking at how that blue line changes between each pair of dashed black lines. But that's really only one component of the total variance, right? That's only the task independent variance. It'd be neat to do that for all of the different movements, and then to align it to session start and see how it changes. So yeah, great idea.
Okay. We have a question from Ralph Hefner.
Yeah, hi, very cool results. I have a question. Do you have a sense for, I know you can't do this with the wide field imaging, but do you have a sense how much neural variability for the spike counts would be left over if you take all these factors into consideration? Are you ending up with something sub-Poisson, or have you thought about this?
Yeah, so that's a good point. We could check that with the NeuroPixels data. I think for sure these observations really bear on how much total spike count variance is due to stochasticity, I suspect. I don't know that, because in experiments that others, including myself, have done, where we measured the Fano factor or related measures over time and found it to fluctuate, we didn't take into account uninstructed movements. And so my hunch is that a lot of that apparent stochasticity is driven by movements that differ idiosyncratically trial to trial. I think already, based on work that I had done as a postdoc, I already feel like the Fano factor is probably way lower than 1, right? It's clear that a lot of what we think of stochasticity is variance that has to do with the sort of task-related variance. And I think now it's clear that a lot of that is probably movement-related variance as well. So like you, I'm super curious about how much stochasticity is left. For sure, some, I guess, right? I mean, we accounted in the wide field for 42% of the variance. That's a lot of variance left over. So probably some of that is movements we didn't track, like breathing and heart rate, but there's still some variance left over. But it's that amount of stochastic variance, I feel like it's getting smaller and smaller as more and more experiments come in. So yeah, I don't know. I'd be curious to know what you think. That topic has for sure been on my mind a lot.
I'm not speculating. It's an empirical question.
Okay, I have a short question. I'm just wondering, you know, you're performing all this with visual stimuli in an animal that probably isn't the best for visual processing. I'm just wondering, you know, when you look instead with another stimulus set, let's say auditory stimuli for them, you're going to see a whole different pattern, I would imagine, of uninstructed movements and other things going on. Is that actually what actually occurs, or do you see the same type of stereotypic movements or whatnot?
Yeah, so in this study, we used both auditory and visual stimuli, and we found, hang on, let me just, I've lost my cursor here. Let me get my cursor back and I'll go to the right plot. We didn't look at, it's an interesting question, actually. I don't know why we didn't do this. It's a good idea. So we can tell the overall amount of variance accounted for by auditory versus visual stimuli. It's a little more with visual stimuli than with auditory stimuli, although probably that's just because we can't image auditory cortex very well. But we haven't really looked at whether there are different patterns of movements that occur following auditory and visual stimuli. That is a great idea, and we could very easily do that. We also do somatosensory trials now as well, although I haven't shown these here. But I think I do think of, you know, when we consider different animals and different modalities, I do have a slightly more nuanced view, I guess, and maybe that came from doing so much work on multisensory integration. Like on the one hand, the spatial acuity of mice is obviously much worse than humans. They use their visual system in a very different way. But you know, they do have at least 7 separate representations of visual space, and they clearly use vision a lot for navigation. And so I guess for me, I started to think about kind of different animals and different modalities in a new way, rather than thinking of, you know, primates as being vision dominated and mice as being like olfactory or whisking dominated. I think that the multisensory view is really helpful, like both in different animals and even in different circumstances, the modalities are differentially reliable, right? Like even for human, if you're talking to someone late in the evening, your visual system is not nearly as useful as it is during the day. Or as a human, you know, if somebody gives you a delicious looking hamburger, but it smells terrible, you're never going to eat it. You know, because your olfactory system is really good at detecting bacteria, and your brain knows about the reliability of that system. So I think we would never want to only use like mice as a model for vision, because aspects of their vision are fundamentally different from human and non-human primates. But I think viewing all animals as being kind of inherently multisensory and in the business of figuring out which modality is the right one at a given moment and for a given task has for me been kind of a helpful way to think about it, inspired partly by, you know, work of Greg DeAngelis and others at Rochester.
You bet. You bet. I'll just ask, you mentioned that there was a slide that you left out, and I'm curious now. I can't forget.
Yeah, I don't know why I did that. That was weird. I highlighted the importance of looking at single cell data and then didn't show anybody the data. So let me just, I skipped those slides for some reason. Okay, so for the, here we go. Okay, so for the single cell data, we did the same thing as we did with the wide field data, except that we're fitting the single cell fluorescence instead of fitting the wide field fluorescence. And so we looked in these 5 different areas. We had pretty good cell counts. We had 12,000 neurons altogether, and we looked in these 5 areas: ALM, MM, V1, retrosplenial, and somatosensory cortex. And in every area, we found that the uninstructed movements dominated. So even when we looked at single cells, rather than this mysterious pooled signal of the wide field, the same conclusions held, that the task and instructed variables made a smaller contribution. That's the summary of the data. And I think I just want to show 3 example cells. Okay, so this is the first cell, and I've plotted this cell 3 times. And you can see that this cell has kind of a nice time varying increase during the stimulus period. And maybe, you know, without knowing anything else about the cell, you think that you might think that this could reflect some computation that you like, maybe movement preparation or integration of evidence. Oops, sorry. And so I'm going to show you now the fit for each of these different model groups for this individual neuron. And so you can see for this individual neuron, it's actually really fit well by the task model. So this is not a movement cell. It was accounted for by a variety of task variables. Okay, so here's another cell. And this cell likewise has a time varying increase that occurs during the stimulus period. That gray line is going up. But this cell is not fit at all by task variables. It's really fit much better by the uninstructed movement model. So I think for me, this cell was really useful because in the past, I would look at a PSTH, like a trial averaged time varying response. And based on that time varying response, I would be tempted to make inferences about the computation that that neuron is reflecting or perhaps performing. But this to me was a real eye opener because you can see two cells that have a pretty similar mean response that reflect utterly different combinations of task, uninstructed, and instructed movement variables. So I think that I highlight this because it's pretty easy these days just to use video. If you don't really care about movements, you can even just take the raw video and reduce its dimensionality, but it's really useful to know the extent to which individual neurons are modulated by movements.
These guys are located right near each other, these two neurons?
I don't know where they were spatially. Yeah, often neighboring neurons have very different, utterly different responses, which is why you don't, for instance, see noise correlations that are through the roof, because neighboring cells can be very different in the extent to which they reflect these parameters.
Sure. Okay, well, if there are no more questions in the audience, and just want to say thank you again for a great talk. And I know some of you are meeting with Anne later, and we'll be getting to have some coffee with the students later on as well. So thanks again. And we really appreciate you coming to University of Rochester.
Thank you so much.